In this video, we are going to discuss lipids under AP Unit 1, Chemistry of Life. Lipids are made up of elements, carbon, hydrogen, and oxygen. They are insoluble in water. This is due to the fact that they have non-polar long chains of hydrocarbons. These long chains of hydrocarbons do not have the tendency to form hydrogen bonds. But what are the four main categories of lipids? The four main categories of lipids are fats, phospholipids, waxes, and steroids. Let's start discussing fats. Fats are formerly known as triglycerides. From their name, triglycerides are made up of three fatty acid molecules that have even number of carbons that range between 16 to 18 carbon atoms. So each one of them is a fatty acid with an even number of carbon ranges between 16 and 18 carbon atoms. But at one terminal, there is a carboxyl functional group that have ability to join the glycerol 3 carbon molecule. This happens by condensation dehydration reaction, by which 3 water molecules will be removed to produce the triglyceride. But what is the main function of triglycerides? Triglycerides are used by animals for insulation and long-term energy storage. But we always discuss triglycerides in terms of being saturated fatty acids and unsaturated fatty acid. Again, fatty acids are these molecules that could be saturated with no double bond. There is no one single double bond found if it is saturated fatty acid. And this is due to the fact that it contains as many hydrogen as they can use. They are saturated. And usually they are linear in their structure, which leaves them solid at room temperature, as in the case of butter. But unfortunately, saturated fatty acids cause many cardiovascular diseases. On the other hand, the unsaturated fatty acids, which often come from a plant source, they have at least one double bond in the and if it is in cis configuration, as we will discuss later, this double bond will leave the fatty acid with a kink. And due to that kink, unsaturated fatty acid has lower melting point compared to saturated fatty acid, which makes them liquid at room temperature. But not all unsaturated fatty acids have this kink due to the double bond. We said only the cis configuration of unsaturated fatty acids have this kink. And what do we mean by cis configuration? Cis configuration means that the hydrogen that is found on the carbon where the double bond is created are at one side, as in this case, or in this case. By mistake here, I didn't draw the kink, but this here, there should be a kink due to the presence of the cis configuration of unsaturated fatty acids. However, due to hydrogenation, a second form or a second configuration of unsaturated fatty acid could be produced, which is the trans fat, and it is considered as a very bad for our health because it increases the bad cholesterol in our body and decreases the good cholesterol in our body. Trans fat means that, or the trans configuration means that the hydrogen on the carbon are alternating. And this kind of trans unsaturated fatty acid is produced for maybe better flavor in junk foods or other uses. But they are known to be linear just as the saturated fatty acid, and they consider to be very harmful for our health as they increase the level of the bad cholesterol and decrease the level of the good cholesterol in our body. However, at the beginning we said that triglycerides or fats, they play an important role not only in insulation but also as a long-term energy storage. But why fats are the long-term energy storage used by animals? This is due to the fact that 1 gram of fat stores more energy than 1 gram of any other biomolecule. But the more important thing is that fat droplets do not contain water. And this is very necessary because take for example small birds. If the fat or if the stored form of energy contains water, this will add more weight to the small bird, which is what will not allow the bird to fly. So having a biomolecule that stores energy without containing water, this will lead to a lower weight for the bird, which allow it to fly easily. But fats are not the only category of lipids. Another category of lipids is phospholipids. Just like triglycerides, phospholipids, they have fatty acid chains and they have glycerol. But the major difference in the structure that there are no 3 fatty acid chains, just 2 fatty acid chains. Unlike triglycerides, as we discussed before, they have 3 fatty acid chains. But another very important difference in structure is the presence of the negatively charged phosphate group. And this makes the head of the phospholipid hydrophilic and polar, while the tails, of course, these long chains of hydrocarbons are hydrophobic. But also we can notice the presence of double bond and the kink, and this is very necessary to make kind of a distance between the 2 fatty acid chains. So phospholipids, they have amphipathic nature. And what do we mean by the word amphipathic? This means that it has a polar head, hydrophilic, thanks for the presence of the negatively charged phosphate group, and non-polar tails, the hydrophobic part. And once phospholipid exposed to water, they have this configuration, or they take a bilayer structure. And this is very important to hide the hydrophobic tails and sandwich them between 2 hydrophilic heads. So these hydrophilic heads are able to be in contact with water, while the hydrophobic tails will be hidden inside the bilayer. Now, the third category in lipids are the steroids. We discussed the phospholipids, we discussed triglycerides. Now, let's talk briefly about steroids. Steroids are lipids with 4 fused rings. And the most, let's say, famous kind of steroids is cholesterol. This is due to the fact that cholesterol play an important role in producing vitamin D. And they also play a role as a component in cell membrane. They provide kind of stability in the cell membrane or the plasma membrane. But also what is very important about cholesterol, that our body can produce testosterone and estrogen using cholesterol. So cholesterol is a precursor for other hormones like testosterone and estrogen. And we can notice from the drawing here that the difference between testosterone and estrogen is just in one of the functional group. So cholesterol as a steroid is important because, as I said, it is a component of cell membrane. It provides stability and a precursor for other steroids as testosterone and estrogen. But the bad cholesterol, the one I mentioned before, the LDL, can contribute to cardiovascular diseases because accumulation or increasing the level of LDL in our body will lead to an accumulation of fatty material in blood vessels, which blocks the flow of blood in it. Now, the last category of lipids are the waxes. Waxes, just like phospholipids and triglycerides, they contain long-chain fatty acids, but these fatty acids are connected to carbon chains that have alcohol function group. This is the unique thing about waxes. They are solid at room temperature, and they play important role in living organisms. For example, in the cuticle, in the leaves, they prevent water loss. They are involved in formation of skin and fur. We can find them as an earwax. Also, bees use them to create its beehive, the bee wax. So this is like briefly about waxes, but what we need to end this video with is the function of lipids. So as discussed earlier, the function of lipids could be structural function, as we talked about cholesterol, for example, that provides stability for the cell membrane. Phospholipids also, they are considered to be a structural component to create this phospholipid bilayer, as will be discussed later. But also lipids, they are source of insulation, as in triglycerides, and they play important role as a long-term energy storage, again as in triglycerides. But also they have signaling, they play signaling roles, or they are signaling molecules, as in testosterone and estrogen, the hormones, and how they signal certain reactions or a cascade of reactions in cells. Now, in the last part of this video, we will be solving one short response question as presented in Barrons. In Barrons, they asked, the question was about a student performing an experiment about two fatty acids, one unsaturated and the other one is saturated. They presented the data here. They showed that the unsaturated fatty acid has a lower melting point compared to saturated fatty acid. The first question was to give one structural difference between these two kinds of fatty acid. One can immediately say that saturated fatty acids have a chain of carbon atoms connected to a single bond. There are no double bonds in saturated fatty acid, while unsaturated fatty acid have carbon chains that contains at least one double bond between the carbon atoms. Then in the second part, they ask about the independent variable and the dependent variable. Of course, the independent variable is the type of fatty acid tested. As we discussed before, the independent variable is the variable that the scientists change. So the scientists change the fatty acid, and the dependent variable is the result. The result is to study the melting point of each one. So the dependent variable is the melting point. In the last part of this question, they said that cell A have unsaturated fatty acids with higher percentage than cell B. So which one have a cell membrane that is more flexible? Of course, cell A. And then they said, prove your answer. One can immediately say that cell A has higher percentage of unsaturated fatty acids, and unsaturated fats contains double bond in random location, causing bends or kinks in the long chain. These kink or these bends in the fatty acid chain prevent unsaturated fatty acid from forming tight packed sheets, which leaves some spaces between the fatty acid chain and make it more let's say flexible.